Okay, the date today is August 8th, 2025. My name is Mike Hogan. I'm currently a postdoc at the University of Michigan. I'm doing research on sensory evolution in snakes. And today, what I'm going to be doing is recording a presentation that I gave at several scientific conferences this summer. And this is just so I can upload it and share it with some people that weren't able to attend the meetings. So my talk is titled Quantifying Pitviper Depth Perception from Preserved Museum Specimens. Before I start, it's really important. I have to give a couple really key shout outs. First to Dr. Allison Davis-Raboski, who I've been working closely with for the past 2 years now. Allison is spearheading a huge effort to CT scan snakes as a way to investigate sensory evolution. And everything I'm sharing today is a smaller offshoot of this larger project. And I also have to give Dr. Tyler Hunt full credit for introducing me to the world of sensory fields. Tyler developed a very elegant approach for measuring crocodilian vision for his PhD. Tyler and I overlapped at Florida State University for graduate school, and so working with Tyler, we basically came up with a way to recreate his analyses, but using the CT data with snakes. So pit vipers are masters of predation, and this is usually attributed to their impressive venom system, which is known for having prey-specific toxicity. Unsurprisingly, there's a huge bias towards venom research in the pit viper literature. But I would argue that pit vipers, what's more compelling about them is that they're masters of perception. After all, the first step in predation is actually perceiving your prey. Traits are optimized through the process of natural selection, and this is in part driven by the coevolution of predator avoidance and prey. Sensory perception must work, so to speak, for a trait like venom to evolve, and I like to illustrate this by saying, if you can't find it, you can't kill it, and you can't eat it, since failure at each point leads to the same fitness response, which in this case is just a hungry snake. My previous work has been on the genomics of sensory perception in pit vipers. In particular, I spent a lot of time characterizing all of the chemosensory genes. This included a huge diversity of vomeral nasal and olfactory type receptors, and over half of this entire diversity were actually all found on the Z sex chromosome. So there's some very interesting genomic architecture going on driving the evolution of sensory perception in these animals. Today, I'm actually going to be taking a step out of the genomics world and focusing more on morphology. And instead of looking at chemoperception, I'm going to be focusing on vision and the thermal sensing ability of pit vipers. So just to give a quick overview of my talk, first, how does depth perception actually work in pit vipers? I'm going to quickly go over what I'll be measuring for that. Next, can sensory fields be recovered from preserved museum specimens? And then finally, what are the factors that predict sensory investment for depth perception? Usually, when we're thinking about depth perception, we're most familiar with vision. And so we have a right eye, which gives us our right monocular field, and then we have a left eye, which gives us our left monocular field. But we also have a region in the middle, which I'm showing here in yellow, where those two monocular fields actually overlap. And this gives us our binocular field. And so by having this overlap, it gives us a way to be able to gauge distance. And so this relationship is precisely what I'll be talking about and what I measured for this research. However, it's not that simple in pit vipers because they also have a thermal-based depth perception. Specifically, their pits are also bifocal, just like the eyes. Their predatory strikes are linked to fitness, and we know that both the eyes and the pits are used for aiming those strikes. However, we don't really know a lot about how much overlap there is between these two sensory inputs. To try to get at this, what I came up with was an approach to model sensory fields for both the eyes and the pits in parallel. I then overlaid the binocular fields together from the eyes and the pits, and I calculated what I'll be calling a quad-ocular field. So this is 4 different inputs going into depth perception for these animals. So just to recap, how does depth perception work in pit vipers? They have bifocal eyes and bifocal pits, and by that I mean there's right and left overlap, and the head shape is also very important for defining their blind spots. So I've been working closely with the University of Michigan Museum of Zoology for close to 2 years now, and I've been incredibly impressed by the quality of research that's going on, specifically on snake evolution. We have a world-class team of scientists in-house. We also have a state-of-the-art CT scanning platform, which includes a dedicated staff who are experts in managing the platform as well as curating the data. And currently, the Museum of Zoology houses the largest preserved snake collection in the world. So this is even larger than the Smithsonian. So putting all these pieces together, we really have a great place to do this sort of research. What I've been trying to focus on is connecting sensory genetics to sensory phenotypes. And so I did a lot of the genetic work for my PhD, and so that's why today I'm going to be focusing just on the morphology side of things. But in the future, my goal is to bridge back to the genetics and form a more complete map of sensory evolution in these animals. So specifically for the CT data, this is the sort of resolution that we're able to achieve. And so I'll just let this video play for a second, and if you watch the top left corner, you'll see the numbers changing as the head rotates. And essentially what I'm doing as the head rotates is counting how many colored pixels there are. So I color-coded the 2 eyes and the 2 pits unique colors, and by tracking how much of those colors are visible for each frame of this video, I'm able to essentially recreate what the limitations are for the sensory fields for both the eyes and for the pits. But crucially, what I'm doing is measuring both of them at the same time. And so you can see I speed up, and we're changing the tilt of the head as we rotate. And you can kind of picture, by mapping out the sensory fields like this, I'm able to recreate full spheres around these head projections. And so this is what I'll actually be using to model these sensory fields. And so you can see the red and the blue corresponding to the right eye and the left eye. And then the yellow part in the middle is the binocular overlap projection onto the sphere. Same thing for the pit. And if I just take those 2 yellow from the eyes and the pits and overlap those, it gives you what I'm showing in the middle. So the yellow that remains is actually that quad-ocular field that I was talking about earlier, where the white above is just the eye binocular field that doesn't overlap, and then the gray below is the pit that doesn't overlap. So just to recap, how can sensory fields be recovered from preserved museum specimens? And I came up with a scaled pixel counting approach as a standardized way to measure this and to be able to compare it between different specimens. Also, the sensory overlap proportions with these spheres allowed me to compare them directly to one another. And so I'll be focusing on the spheres for all of my analyses. For the rest of my talk, I'm going to be talking about these 21 different pit viper species. You can see I have a really good spread of old world and new world pit vipers. And this diversity also includes a pretty decent distribution of different lifestyles and also diets. For the sake of my talk, I'm just going to be focusing on whether it's an arboreal species or terrestrial species. And for the diet, the one takeaway I want to point out is whether something is a mammal specialist. So that means it only eats mammals. I'm going to point out one of the results later that correspond with that. And just as a reminder, what I'm actually comparing are these sphere projections for each of these CT scans. And so you can see the eye field and the pit field, and then the binocular overlap field. Okay, so for my first main result, I found evidence for sensory trade-off. And in particular, what I found is that the relationship of the eye binocular field that overlaps with the quadocular field transitions to having a large eye binocular field that doesn't overlap to a large pit field that doesn't overlap. And so, for example, what I did to illustrate this is I pulled out two extremes from this plot here. And if you compare both of them, so if you look on the right side and you compare the two projections, you can see that the quadocular field is a similar size between these two. So it seems like it's important to have a certain amount of binocular field from either the eye or the pit that doesn't overlap. This is just an interesting result, and it's an example of sensory trade-off with shifting binocular fields based on different head shapes. For my second result, I found evidence for an ecological bias from diet. Specifically, what I found is that there is a significant difference in the angle that the fields are actually pointing in. So we're just focusing on the quadocular field here. So just that, overlap between the eyes and the pits. And what I found is that the direction that quadocular field is pointing is slightly higher for specialists. And I found that the relationship is even stronger when I just focus on mammal specialists. And since we're talking about degrees, I think this would actually be a little bit easier to visualize to show it in this sort of format. And so what I have here is completely horizontal flat is 0 degrees. That's just in line with the head. And all of these arrows represent all my different specimens and what direction, the average direction that the quadocular field is pointing at. And so if I just color in my mammal specialists, you can see that they all occur bunched up at the top, either inline horizontal or above that horizontal origin. And so this is an interesting result, and it suggests that the angle of the actual quadocular field is ecologically relevant. And for my third result, this is just focusing on the total visual field. So for this one, ignoring the pits. What I found is that arboreal pit vipers have the largest visual fields. And this actually makes a lot of sense if you look at the shapes of the head and also think about where the snakes are doing their hunting, and obviously they need to look down. There's more of a three-dimensional sensory input that they need to process to be able to be successful predators in that sort of environment. And so I have 3 example arboreal pit viper heads, and for all of these you can actually see both red and blue pixels from directly below the heads. And to demonstrate this, I actually have some 3D prints. So this is a 3D print of an eastern diamondback head, and you can see I have a light on the inside of it. You can see when I lift it directly up, you can't see the eyes. Now, I also have this Tropodolamus 3D print, and you can see when I hold it directly facing upwards, you can still see the red and the blue from the pupils of this snake head. And so putatively, this means that there is some depth perception information that's available to the snake from directly below their head when they're climbing in the trees. and the rattlesnake does not have that ability. So to recap, what are the factors that predict sensory investment for depth perception in pit vipers? And I found evidence for sensory trade-off between the eyes and the pits. I also found that mammal specialists have a more upward pointed depth perception, focusing on that quadocular field. And also arboreal snakes have the largest visual fields. And so that's the bulk of my talk today. But just to finish, I wanted to try and look beyond skin deep anatomy. So there's a newish micro CT scanning approach called DICE-CT. DICE stands for diffusible iodine contrast enhanced CT. And effectively what this does is it dissolves iodine into the specimen, and different tissues absorb iodine at different rates. What this does is it gives us an incredibly high contrast of different tissue types based on how much iodine is absorbed. If you've ever been to the doctors and had blood work done and needed a CT scan, you probably had some sort of iodine injected into your bloodstream. The methods for DICE staining museum specimens was actually published by our very own Dr. Haley Crowell, who recently completed her PhD here at the University of Michigan. So I definitely recommend checking out her paper if you want more details on how to do this. But just to illustrate how powerful this technique can be, I'm just going to play this video. This is a Crotalus pricei. It's a twin spot rattlesnake, and we're basically going to pass through the body from 3 different perspectives. And so first, from the top of the head going down, you can see inside the skull, you can see the brain, the 2 eyes, the nasal passageway, all of that muscle that surrounds the head, and then the trachea going down there. And then now for this next pass, we're going to go through the side of the head. And so we'll be able to see the venom gland, the right venom gland, almost immediately right here. You can see the muscle stains much brighter than the venom gland. And so there's the eye and the retina. You can also see the inner ear, the nasal passageway, the olfactory bulb. There's the trachea again. More musculature, and here's the left venom gland. And again, this is all without having to cut open the animal, and we're able to retrieve this incredibly detailed information. And so now, starting at the nose, we're going to pass through the body. And so here is the nasal passageway, and then those 2 lobes were the olfactory bulb. We're about to see the eyes, so the lenses stain very bright, so those are the lenses. And we can also see the tongue towards the bottom, those 2 brighter muscles that are going down further into the body. We got the right and the left venom gland with the muscles surrounding it. And so getting that soft tissue anatomy really opens the door to new areas that we can begin to look. And with that, I would just like to thank my funding through the National Science Foundation. Also, I have to thank Dr. Allison Davis-Roboski and her amazing lab. Lots of awesome people at the museum who I've worked with, Ramon, Haley. So here's just some key tools, and please email me if you have any questions. Thank you for watching.